Hello, this is Austin in Fiji. I'm with Corals for Conservation. I've been working with coral restoration for over 30 years now. I've been learning lessons in the Caribbean and the Pacific, working in at least a dozen countries over the years. We have a crisis now with coral reefs. Coral reefs are in the forefront of a global collapse due to climate change. And I'm re-examining everything that I've learned over these years and trying to come together with a new coral-focused model for climate change adaptation and restoration, new strategies that can help save these coral reefs from impending extinction. And this presentation will basically show you 30 years of learning and struggles and tips on how we can move forward. So we need to save these coral reefs, and we need your help. Many of you have seen the BBC footage of our corals that was filmed in 2009. And all of the corals on that beautiful footage, showing them growing in fast motion, died from bleaching. This was very discouraging. We had involved the community in the site. They had established no fishing areas on the reef. We had done giant clam restocking. It was really very exciting. And when these corals all died, it was very discouraging to me and to all the people in the community. And it basically caused the whole project to fall apart. So 5 years of work with the communities and on the reef, all gone. Thousands of corals dead. And what did I learn? This is the most important question to ask yourself. Will the corals that you're working with, will they survive for the next 20 or 30 years? If you don't ask that question now, then my answer is that they probably won't. Let's look at Kiribati. This is Christmas Island, the biggest atoll in the world as far as land area. In 2015 and 16, a mass bleaching hit there that lasted over 10 months. 95% of the corals died. And even though they had these beautiful corals inside the lagoon that could survive to about 35 degrees, all those corals died. People had said, oh, those are heat adapted, they're pre-adapted, they're going to survive okay with climate change. But what happened was, when the ocean became 31 and 32, the lagoon became even hotter, and so they died. Just because a coral is heat adapted does not mean it can survive where it is now. We searched for 4 days, and we couldn't find any corals in the lagoon. Very discouraging. Walking on the beach of Christmas Atoll, you'll find all of these tabulate corals all over the beach. And right now, today, you cannot find a single tabulate coral growing on this atoll. They are extinct on this atoll. This is the only colony that I found alive of the Acropora. This is Acropora globiceps. It's a digitate colony, and only a little bit of the colony was alive. So Kiribati is our future. It's the front line of the collapse of coral reef systems all over the world. It tells us where we are going in the coming decades. Severe bleaching temperatures for half the year, every year, except for La Nina conditions. And in Kiribati, only 5 of their 33 atolls have even been surveyed since the disaster 7 years ago. The 3 columns on the left, the Gilbert, the Phoenix, and the Line Islands, all make up Kiribati, the nation of Kiribati. They are the leaders in the collapse. You can see how they are the most severely bleached of all the reefs. The Maldive Islands, the little red column, it bleached badly in 1998, and most of the corals died. And they were awarded $100 million from the United Nations in compensation. Kiribati has not received a single dollar. These are the lessons that I've learned by working at the front line of the coral reef collapse in Kiribati. That the heat-adapted coral populations of hot pockets are just as vulnerable because they exist at the upper limit of thermal tolerance. That without translocation, hot pocket corals are going to die. Acropora species are particularly vulnerable, and they're the first ones that face extinction. When acropora corals become rare, they can be aggressively devoured by parrotfish or other predators. Acropora dominance is often replaced by Pocillopora dominance, and then in turn it gives way to Porites dominance as the stress continues. However, the Global Coral Reef Monitoring Network will not show that. It only uses coral cover, and so it's a poor indicator of these phase shifts. So we need more fine-tuned measures at genus level. So what are the strategies that are already operational to save coral reefs from extinction due to climate change? There's only one that's been widely recognized, and that's the 50 Reefs Initiative. And this strategy uses modeling to select from all the reefs in the world the 50 reefs that are most likely to survive into the future, so that we can devote our resources to saving those reefs. It doesn't support highly impacted coral reefs like Kiribati, and it does not have any coral-focused or restoration components. So we are proposing Reefs of Hope, which is a coral-focused strategy, and it focuses on the pre-adapted corals that are resistant to bleaching, resistant to acidification, and resistant to disease. These are the three things that the nearshore hot pockets have been selecting for over hundreds of years, thousands of years, okay? And we translocate those corals, and we use them to focus on ecosystem-based restoration and giving hope for the future, because those cool water reefs are the ones that are severely impacted by bleaching, and they're dying out. So these are the same species that were originally found on those cool outer reefs, and we're getting them restored so they can reboot natural process, natural reproduction, natural larval recruitment, and that sort of thing. More on the 50 Reefs Initiative. The map at the top of this slide shows the blue reefs which have been selected as the most likely of surviving, and the red reefs which have been selected as the least likely of surviving, based upon this 50 Reefs Initiative model. The United Nations has selected this strategy, the 50 Reefs Initiative, as their basis of making decisions. And so the Global Fund for Coral Reefs, they have project sites only in the blue or green areas, and the red areas need not apply. So because this model is influencing major decisions right now, we need to look at that model and look at the parameters they've chosen to make this determination, which then affects everybody in those countries. So if you look at the 5 parameters, 3 of them are basically overlapping. They are related to thermal stress and thermal history and projected thermal conditions, while only 1/3 of the model is related to anything else. Where is ocean acidification in the model? Where is the present condition of the reefs and getting things under control, like overfishing and land-based threats? So if you look at the Caribbean, Belize, which has really the least threats and the best reefs, is ranked red or orange, and Hispaniola, with the worst reefs in the Caribbean, some of the worst reefs and huge stresses from land and overfishing, is somehow blue. So there's something not working exactly right with this model. If you look at cyclones, which is the purple pie section here, the cyclones are considered in the model as totally negative. But based upon our experience, yes, cyclones can be negative. They wipe out maybe a 10-kilometer section of reefs, but then a 1,000-kilometer section is cooled off and protected, and it prevents bleaching in a wide area. So when NOAA predicts bleaching is coming, and we're all panicked, oh no, mass bleaching is coming, then often, most of the time, there will be tropical storms form or cyclones, and they'll go near us, but not on us, and they cool off our site. So the last 5 or 6 predictions of mass bleaching have backed right off due to cyclones. So cyclones should either be taken out of the model or made as a positive influence on many reefs. So you see, the model needs to be looked at, and it's good to have models, but those models need to be flexible, and we need to update the model now. So this is how far we've gotten with our Reefs of Hope initiative. We have our training sites established in Fiji, good model sites, and we've also done international training. We've done training in Vanuatu, Tuvalu, Samoa, Kiribati, and French Polynesia. And we've had trainees come from Papua New Guinea and New Caledonia and various other places in the region as well. So we have a foothold started, and we have a good concept, and over 100 people trained. And these are our Fiji Reefs of Hope sites. Our primary site is in Malolo in the west of Fiji, not far from Nadi. And we have extension sites that we're developing as well. So Reefs of Hope is doing the best we can, and we are learning lessons along the way. This is adaptation. It's also ecologically based restoration, ecosystem restoration. And we've learned certain lessons that we feel need to be shared with the wider community. I'm going to tell you some of those lessons now. Lesson 1. After bleaching is over, predation can eliminate the coral survivors. So you've had your mass bleaching, the waters are cool, and maybe 10% of your corals have survived, of your acroporas. And you think, well, that's great. We have some really strong corals here. The reef is going to learn how to adapt. But the problem is that doesn't happen, because left alone, the predators on that reef will eliminate virtually everything that's left within months. This is our experience. So what happens after a bleaching? The predator to prey ratio is skewed. And what was a very rare predator with respect to the amount of corals for them to eat has now become super abundant. So in the Pacific, the crown of thorn starfish will eliminate most of everything. And then the Drupella snails will finish it off. So this is extremely important, and this is something that we need to address if we want to help corals to adapt. Now, the Caribbean, you have different species, but you have the same sorts of problems where predation can eliminate what survives when corals become quite rare. Even these beautiful butterfly fish can destroy the corals. When a cropper becomes rare, they strip all the tissues off. It's pretty amazing. Parrotfish, oh my goodness. Parrotfish are supposed to be great, but they will turn on your corals. Some species, they love to eat live corals. They will gobble them up. One of the species that really helps to keep coral alive, a cropper especially, on the reef are these Stegastes farmer fish, these damselfish. They grow algae in the bases of the corals, and they do damage the corals a little bit on the bottom, but these are keeping the corals from being killed out by the predators. They can chase the crown of thorns away. The snails don't seem to be able to get in these corals. The parrotfish will be chased away. The butterfly fish will be chased away. So these fish are extremely important, and I believe they are critical to the adaptation on coral reefs because they keep cropper corals alive when a cropper becomes very rare. Okay, so lesson 2, we establish resistant corals within gene bank nurseries where they are safe from most predators, okay? So here's one of our gene bank nurseries in Fiji. The crown of thorns can't find them. The snails can't find them. This is our major site in Fiji, in the Mamanuca Islands in Malolo District. It is a natural leeward reef, and it is a natural hot area. So it's good for stress, and we are in a place that is naturally stressful for good reason. The Mamanuca Islands have a fairly unique double barrier reef system. We have fringing reefs up against the islands. Then we have the middle barrier reef and a cool outer barrier reef on the outside. The cool outer barrier reef, the entire inner reef, the back reef, is mostly dead. These corals were not well adapted, and there have been major cyclones as well hitting this reef. So we have the cool reefs, we have the moderately warm reefs, and we have the very hot reefs. So lesson 3 is we're translocating the thermally adapted hot pocket corals from these inner reefs to the middle reefs, and from the middle reefs to the outer reefs. And this is our attempt to secure the corals from more severe bleaching events because they are coming. This is Nuku Reef. These corals are in the middle barrier reef. They are the same species that are found on the outer barrier reef in the cool, but these are heat adapted. They can take 32 degrees without major bleaching and without coral death. Within the hot pockets, although a lot of those areas are dominated by porites, there are still some acropora corals, and often located within farmer fish territories. So we will get these colonies out, sometimes sections or even entire colonies if they aren't too big, and we will sample from them and take them out and cement them onto these biscuits or cookies, and then put them into the nursery. And we start with testing nurseries to test them to make sure that they are strong before we graduate them out to the outer reef. And this is one of our testing nurseries in the nearshore waters. These corals have mostly been graduated and taken back out to the cooler nurseries now. This is a stress test nursery in the near shore, and you can see how two of the genotypes, they've been growing for several months, and they're bleaching, the two on the left. So these two genotypes will be retired and put somewhere on a cool reef, and we won't use them anymore. The two on the right, they are indeed unbleached. They are supercorals. They're surviving and thriving in 34, 35 degree water. And these are worthy of growing up large and using them in restoration sites. Okay, lastly, coral reefs are the rainforests of the sea. So why are we using methods modeled after commercial forests and timber production? Tropical forest restoration models actually now involve fewer and larger transplants clustered together. So they take larger saplings several meters high, which will attract birds because the birds can perch on them. And then they plant dense patches of those trees, which amplify success via habitat creation. So we're trying to attract the wildlife. And that wildlife, the birds in particular, remove predators, the insects, they increase the nutrient flow, and they plant new species. So on our reefs, fish are the birds. And by planting larger branches closer together, we will reboot some natural processes, and this is what we're doing. So not only are we proposing a new paradigm for climate change adaptation, we're also proposing a new model for restoration, whereby we use larger colonies, entire colonies of corals, and stop using these tiny little nubbins that we've always used. Because we want to jumpstart natural processes that are based upon the fish cleaning the substrate, the fish eating the algae, the fish protecting the corals. One of the colonies on each of these ropes we will trim, and we will create a new rope that will replace it in the nursery. We do not want to lose any of our genotypes. After we trim one colony per genotype rope and replant the genotype within the gene bank nursery, we move the bulk of the coral, these large colonies, out to the outplanting site, where we create nucleation patches on this outer cooler reef. What we're doing is we're creating instant fish habitat, so the fish move in and start making changes on the degraded reef. Now you'll see that the corals are directly on the boat, and we're spraying them and sprinkling them. We've gone now to spray bottles. It provides a mist, and there's absolutely no mortality when we transport them gently in this manner. So we're moving these large adult-sized corals out to the outer reef, and we have multiple genotypes in each nucleation patch. So coral spawning will happen, and we'll have lots of larvae being formed. But that's not our main point. The main point is to attract abundant fish in these large transplants. And that will clean the substrate and improve the settlement surface for coral larvae. So that's what we're trying to do, is have the fish clean the surrounding area. Now, the acropora corals that we're planting also produced a short-lived pheromone. It's a smell that's proven to be a strong settlement signal for coral larvae. And it helps the coral larvae find the reef. And so it accelerates these larval-based recovery processes. So the newly settled acropora larvae, they come in without their symbiotic algae, and they acquire their algae from what leaks out from nearby corals. So since the coral larvae are settling near these supercorals, they will most likely acquire super symbionts, the super algae. So they're becoming supercorals. And so it looks like that this is contagious, that superness is a contagious factor. And also, the corals that are coming in are of different species. So the diversity of the patch increases, as well as the resilience of that patch to future climate change scenarios. So there may be additional sharing between adult colonies. We're not sure about that, but we think it might be possible and potentially mediated by butterfly fish biting and going between colonies. Butterfly fish spread disease, and they also might be spreading some of the super algae. And we cannot forget that if people are the problem in this world, then people are also the solution. So we need to look at solutions that are community appropriate, that are appropriate for implementation by the tourism industry, by the fishing communities that are dependent on those very reefs. So in the Pacific Islands, we are starting Reefs of Hope, and we have community-based components. We're trying to nest this within existing strategies of community-based no fishing areas and that sort of thing. So let's all get up and let's do our best to help save these reefs. Vinaka Vakalevu, thank you very much to all the partners and donors that have helped over these last few years. And a special loving memory for Taratau Kirata, who just passed away in May. He was really the one who facilitated the work in Christmas Island in Kiribati. Thank you.